Hi everyone, my name is Pranay, and in this session we are going to discuss dynamic causal modeling for cross-spectral densities or DCM for CSD. At its core, DCM is about answering a simple but very powerful question. What kind of neural system could have generated the signals we observe? And to answer that, we need two ingredients: a generative model that tells us how latent neural dynamics give rise to signal, and a way to invert that model using data to infer the most likely hidden causes, such as strengths of certain connections or synaptic properties. DCM is such a Bayesian framework. The forward model problem is modeling how hidden parameters generate data, while the inverse problem is inferring the posterior distributions of parameters from the data we have actually observed. In this talk, we'll focus on how this plays out when the data we are trying to explain are spectral features from EEG, MEG, or LFP recordings. So I'll first expand on the DCM framework. We'll then look at spectral features, how they're computed, and what they capture about brain signals. From there, we'll proceed to introduce neural mass models, which give us a biologically grounded way to describe population level neural dynamics. We'll see how these models generate predicted spectra. And once we have a generative model for spectra, we'll look at how DCM fits these models to data. And using that, how we can make inferences about the model structure or any hidden neural parameters. And then we'll end with a short summary. The generative model in DCM consists of two pieces. The first is a neuronal model, which consists of neural state equations that describe the dynamics of brain activity. Now, these are essentially coupled differential equations that describe how neural circuits interact and evolve over time. The output of this circuit is usually the depolarization of pyramidal neurons. And it is this activity that is treated as the activity of a cortical source, which could be electrical activity or changes in the magnetic field. The observation model takes this activity and maps it to the observations at our sensors. Now, in the case of EEG or MEG, this forward model is the lead field. For LFP, it could be something as simple as a gain function, a scalar gain function. So the generative model in DCM, comprising of these two subcomponents, um, offers a way to link observed data features with hypothesized mechanisms. The forward direction of modeling asks what measurements of brain activity does the model predict given changes in some parameters. Whereas, and this is something we are more interested in, is model inversion, which is what parameters of the model best explain any observed measurements. In DCM for CSD, the observed data is cross-spectral densities. So now, what are spectral features? The Fourier theorem says that any signal in the time domain can be expressed as a sum of complex valued sinusoids. This is the frequency domain representation, which consists of different sinusoids, which can vary in their frequency, in their amplitude, and in their offset, in their shift. Formally, this is expressed in this equation where small x of t is the time domain representation of a signal, and capital X, which is a function of frequency now, is the frequency domain representation of that signal. The third term is the complex sinusoid with a certain frequency. Now, using this expression, We can compute the amplitude at each frequency. We can compute the phase of each frequency, which is the complex argument. And we can also compute the power spectrum, which is the square of the amplitude. Now, from one signal, we can estimate the power spectral density, like I just described. What we can also do is we can compute its autocorrelation, which is the average of this signal convolved with lags of this same signal, right? Now, the Wiener-Khinchin theorem says that the power spectral density under certain assumptions of stationarity is the same as the Fourier transform of the autocorrelation. So that gives us another way to look at power spectral density. Now with two signals, X and Y, we can now compute its cross correlation. So uh, the convolving one signal with lagged versions of the other. Right? Now, if we take the Fourier transform of this cross correlation, we obtain what is called the cross spectral density. It is defined this way. This cross spectral density contains information about how these two signals, X and Y, are coupled at different frequencies. So any shared fluctuations at different frequencies. Right, so this is what it looks like. We have a signal X, a signal Y. We compute its power spectrum density. We also compute its cross spectral density, which now says at each frequency, what is the strength of shared fluctuations. Now, the question is, what kind of underlying mechanisms in a system could give rise to these observed spectra? And we can start off with a simpler case where we have a linear time invariant system whose dynamics are expressed as these two equations. The first equation, x dot, is the rate of change of x. So dx by dt, this is a compact representation, x dot, is a linear combination of ax plus bu. So x is the current state, u is an external input, a and B capture the dynamics. So A captures the intrinsic dynamics of this system, and B captures the dynamics that are due to the input drive, right? Y is sort of the observation that is obtained by applying a mapping to these different X states. So X is all the hidden states in our system, and C transforms these hidden states into the observation Y. Right, now we could apply a Fourier transform to this to obtain this particular equation, which has j2 pi f multiplied. This is because differentiation in the time domain corresponds to multiplying by this term in the frequency domain. So now this expression is a function of frequencies. We can simplify and rearrange to get this in terms of y. And we can see that y is a function of this big term here and the input, right? And this big term is the transfer function. The transfer function essentially relates the input and the output spectra. And if you see the terms in it, it makes no reference to the hidden states x. It only depends on the parameters, and these parameters are C, A, and B, in addition to the frequency F. So once we have this short formulation, compact formulation of Y in terms of the transfer function and the input, we can again compute the power spectrum and the cross spectrum. And again, both of these depend only on the transfer function and the input, not the hidden states. So what do we have so far? We can represent signals as a sum of oscillations using the Fourier transform. This is an orthogonal projection. The PSD captures how signal power is distributed across frequencies for a signal. Cross-spectral density takes two signals and describes the shared fluctuations between these two signals across frequencies. We also saw that a simple dynamical system can generate output spectra through a transfer function, which depends only on the parameters and not the states. In DCM for CSD, our goal is to go beyond just describing the spectra and to explain these observed spectral features using underlying neuronal mechanisms. So in order to do that, we need a model of how neural activity generates spectra, which brings us to neural mass models. Now these sorts of models have had a long history, a rich history, and the earliest attempts were to model generators of the alpha rhythm on scalp EEG. Specifically, Lopez de Silva in 1974 modeled the alpha activity generated in the thalamus. He used this coupled dynamical system consisting of two populations, a thalamic relay cell and an inhibitory interneuron. Now, he used this to successfully model the alpha rhythm and the power spectra generated by the cortex of a dog. Okay, it's a reasonable fit. Now, what is uh, this model comprised of? So we can start by modeling the neocortex, which consists of these 6 layers. Specifically, we can look at one unit or one column of the cortex. Now, if you see this layer 4 consists of these granular patches, and hence it's called the granular layer. This granular layer consists of spiny stellate cells, which look like this. They're star-shaped, hence stellate. These are glutamatergic neurons. They receive inputs from thalamus and distant cortical areas. The layers below the granular layer, so layer 5 and layer 6, the infragranular layers, consists of pyramidal neurons. And these pyramidal neurons have dendrites that span the top layers. The depolarization of these pyramidal neurons is treated as the output of this cortical column. Similarly, the top 3 layers, the supragranular layers, consist of a variety of cells, but for simplification, we focus on the GABAergic inhibitory interneurons, which are densely found in the layer 2 and layer 3. So we have sort of simplified this 6-layer cortex with numerous cell types into just 3 populations: the inhibitory interneurons, spiny stellate, and pyramidal population. This 3-population model with certain sort of connections that constitute a circuit is called the Janssen-Ritt model. It was described in 1995 by Janssen and Ritt and was soon absorbed into DCM as the first neuronal model. So this is the structure of the model. What does the model do? How does it work? For that, we need to describe the equations that govern its behavior. So let's look at the spiny stellate population. This population receives an external input. Now, the change in the voltage of this population is given by as a function of current, and the change in the current is a function of input, current, and voltage. Well, a linear combination of these. And h and tau are just scalar constants. And they are parameters, essentially, that we are usually interested in. H is the average synaptic gain, and tau is a dendritic time constant. I'll come back to this in a bit. Now, the solution to this coupled first-order differential equation is this integral. And this integral is sort of equivalently expressed as a convolution operator between, and this is the convolution between the input u and h, which is an impulse response, which is given by this equation. And this is what this impulse response looks like. This impulse is essentially the postsynaptic potential elicited by a single spike input. And it has parameters. So tau is the dendritic time constant, which controls the decay of the postsynaptic potential. And it reflects the distance, transmission distance from dendrites, which is where the input is received, all the way to soma. A smaller time constant reflects faster dynamics, so the impulse response will rise faster and decay faster. whereas a larger time constant reflects a slower damping, so slower rise and slower fall. H is the average synaptic gain, which controls the height of this postsynaptic potential. Right. So now the spiny stellate cell population receives um, inputs from the pyramidal population, and this is scaled by a parameter gamma, and gamma reflects the intrinsic connection strength, and basically it's the average number of synaptic contacts made by the pyramidal population onto the stellate pre postsynaptic membrane. Right. So yes. Now, in addition to introducing this parameter, we've also introduced another operator called the potential to rate operator, S. This transforms the average depolarization of the pyramidal population to a firing rate that is received by the spiny stellate population. And this is usually modeled as a sigmoid. Um, and this sigmoid essentially controls the ceiling, saturation ceiling for the firing rate. And it's controlled by an offset parameter and a parameter R, which controls the slope of the firing rate. The slope controls sensitivity to voltage, whether the firing rate will climb very rapidly or it will slowly climb in response to increase in voltage. Right. So now what is happening in each population? There's a certain sequence where each population receives presynaptic input, which is in the form of spikes. This input is converted to a postsynaptic membrane potential using the rate to potential operator, which convolves the input spikes with a synaptic kernel. Now, this membrane potential is now converted to a firing rate by the sigmoid transform or the potential to rate operator. Right, so this is one population. If we look at two population, it's this average firing rate from one population is fed to another population where the same process goes through. Right. Now coming back to our equations, we can write the same equations down for the inhibitory interneuronal population, which only receives one input from the pyramidal population scaled by gamma 3. And for pyramidal population, we see there's one set of connections which are depolarization due to the excitatory connections from the spiny stellate, and there's another set which is inhibitory and thus hyperpolarizing. So the net depolarization of the pyramidal population is just the difference between those two. Right. So what we have now is the ERP model in DCM, which is constrained to have these three parameters. So the two dendritic time constants, excitatory and inhibitory, these are shared wherever shared between populations. H is the synaptic gain. Again, these are shared parameters. And the four lambda values are the intrinsic coupling strengths, which are derived from biological estimates. Now, in the original paper that introduced this model in 2003, it was a very fascinating sort of simulation. They took the two time constants, the excitatory on the x-axis and the inhibitory on the y-axis, and they varied it systematically. And they found that the system is capable of producing a diverse range of spectral responses in different bands, of course. So for very fast or smaller values of these time constants, which reflects faster kinetics, faster dynamics, we see activities dominantly in the gamma band, and that's what the time series look like. And as we slow down these time constants, we start seeing slower and slower dynamics in the time courses. Now, these dynamics are even richer if each of the populations receive extrinsic connections from other cortical sources. So that brings us to the question of how do we connect two cortical columns? In 1991, Fellman and Van Essen studied a lot of different cortical microcircuits and came up with these three different motifs of how two regions in various hierarchies are organized. So these are the bottom up, top down, and lateral connections. In DCM, um, let's say we have two regions, one lower in the hierarchy, one further up in the hierarchy of cortical processing. The output of the lower region is fed to the spiny stellate population of the higher region. This is called a forward connection, reflecting a sort of feed forward information processing. The higher region can now do its own computation and feed its output is fed back into the lower region, which goes into the inhibitory interneuronal population and the pyramidal population. This is called a backward connection. So these are the two types. The third type is lateral connections. So let's say we have two regions which are sort of homologous. They are left and right pairs in the hemispheres. So they don't necessarily occupy a hierarchy. So these would be connected by lateral connections. And these lateral connections go to all three layers. And often these are bilaterally connected, so they're bidirectional. Right. So now we need to update our equations to accommodate these extrinsic connections in addition to these intrinsic connections that we saw earlier. And that's done by adding them to the output, sorry, the input of the pyramidal population. And that's what that looks like. So for the spiny stellate cell population, which receives forward and lateral connections, these are the two additions. Similarly, for inhibitory interneurons, we get which receives inputs from backward and lateral connections. These are the additions. And for pyramidal, it's backward and lateral connections, which are included here in the equations. Right. So now this is the full ERP model, a three population convolution based Janssen-Rit architecture with extrinsic connectivity between multiple regions or nodes. Now, how does this neural mass model produce spectra in response to some input fluctuations? For that, we go back to our linear time invariant system, which had this nice linear form. Our neural mass model uh, features a lot of nonlinearities. So one being the sigmoid transformation, which converts membrane potential to firing rates. Now, this is a compact representation of our neural mass model, where x dot is the sort of concatenated or stacked well, all the equations, all the 9 differential equations that we saw for those three populations in total, they're all stacked into this. And it's called the fx function, which describes the latent neuronal states. And this function is uh, this essentially this function depends on the current state, the input, and parameters theta. Y is the observation model, which we haven't yet spoken about, but we'll come to it later on. And it's usually a linear model. Right, so now we can assume that this neural mass model is operating at steady state. And we can use this knowledge to, or this assumption to linearize this model at or around the steady state. We can do this using a first order Taylor expansion. And by doing this, the zero order terms x0 and u0 will disappear because the system is at the steady state, and we are left with this form, which very much resembles the LTI system. And we can hence go forth and do the same sort of derivation to arrive at the transfer function for this linearized neural mass model. We first apply a Fourier transform. Then we solve for the output y by rearranging terms and doing algebra. And we arrive at this form again, where now the output spectra y is a function of the transfer function and the input. The transfer function itself is a function of just the parameters and not the states. Right. So now once we have this compact form where the output spectra is described entirely by the transfer function and the input noise spectrum, we can go ahead and compute the power spectra as well as the cross spectra between two channels or two sources. The observed spectra, both PSD and CSD, are shaped by how the input fluctuations are filtered through the system's transfer functions. The input is usually specified as Gaussian or colored noise, which follows a 1 over f power law. And another way to see this sort of processing is that the linearized neural mass model acts as a filter and hence shapes this input noise spectrum into an output through the transfer function. And the transfer function depends on the system's parameters theta only. Now, in the original paper that described DCM for cross-spectral densities by Rosalind Moran in 2007, there's this nice illustration where the effects of various parameters on the transfer function were demonstrated. So, for instance, here is the excitatory time constant, and increasing this excitatory time constant causes sort of broadband changes, where the most pronounced effects are on the delta and sort of theta band, where increasing the time constant dramatically increases the magnitude to which these frequencies are present. Similarly, the excitatory synaptic gain also produces a similar effect in the lower frequencies, but it has a pronounced effect on a very narrow range of frequencies from 16 to 20 hertz, which rise quite steeply. Right. So now I'll briefly touch upon the various sorts of neuronal models in DCM. The one we just saw is the ERP model. There are two variations of it, the LFP and the SEP model, both of which have similar architectures, but the parameterization is slightly different to produce faster dynamics. Now, the way this model is constructed is by assuming that each population has, let's say, 1,000 or so neurons, and we lump all the parameters of these 1,000 neurons into one by averaging, and we treat just the average as reflecting the whole population. So this is called the point mass reduction. Another way to go about this is by preserving the variability across populations, across neurons, and treating that the cortical sheet as a continuous neural field in two dimensions. So this preserves variability within a population across different neurons. These kinds of models are much more complex. They're called neural field models, and they are implemented in DCM, and they're much more challenging to use. Another way to look at this classification of models is how current is modeled. Now, there is another class of models where the neuronal activity can be due to conductance through a certain set of channels, such as AMPA, GABA, and NMDA. And it is current through these conductance that then changes membrane potential. Right. So more concretely, current I is a function of G, which is conductance, and membrane potential. So this is multiplicative, and this introduces a lot more complex nonlinearities. Whereas the previous models that we've seen so far, they were convolution based, where the current was essentially, it had a linear form. So these conductance-based models were introduced in 2008 by Maria Roche, and this model is called the NMM, which features conductance of AMPA and GABA receptors. There was subsequently a variant of this which also had the NMDA receptor and its conductances, and both of these have their field variants as well. More recently, well, a decade ago, the pyramidal population was split, and this led to refinements where now the model consists of four populations. The spiny stellate and the inhibitory neurons are the same, but the pyramidal population is split into a superficial population and a deep pyramidal population. This was done by Bastos in 2012, and this refined model is called the canonical microcircuit model. It is a much more realistic biological circuit, and is very useful in modeling cortical processing as it happens in a sort of predictive coding hierarchy. Now, there are conductance-based variants of this called the CMM and the CMM NMDA. CMM features, again, AMPA and GABA receptor conductances, whereas the CMM NMDA one, like the name suggests, adds on the NMDA receptor um, conductances. This has seen much more use in the past several years. And one very nice example was in this review article by Ines Pereira, which used this CMM NMDA model. So this is what it looks like. There are four populations. They're connected a certain way. The black arrows indicate all the excitatory glutamatergic connections, which release glutamate and act on the AMPA and NMDA receptors, whereas the red ones are inhibitory and act on GABA receptors. So this review paper is an excellent tutorial, and they use the CMM and MDA to describe cross-power spectral densities. And they have some interesting simulations, which I'll show you. So for instance, this is the power spectrum generated by the system. And there's one parameter that's being varied. This is a simulation, it's a forward model. And the parameter that's being varied is the rate constant of AMPA receptors. This rate constant controls how quickly or slowly the conductance will rise and fall in response to an input, right? And faster rate constant, so increasing red here, a faster rate constant essentially allows less conductance to sort of rise and well, less current to flow through, and hence the contribution is decreased, whereas a slower rate allows a greater net current to flow through, and that leads to a larger magnitude at a certain frequency. There's also a slight frequency shift, but it's very small. Right. The other parameter they varied many parameters, but here is another one, which is the connection strength from the spiny stellate population to the superficial pyramidal population. This is excitatory. And if this connection strength is increased, there is a sort of broadband increase in power, but the predominant effect is in the lower range between delta and theta bands. Right. So a summary of what we've done so far. We've built a neural mass model. So namely the Janssen-Dritt model, which was one of the first models to be included in DCM. We saw how this model can be linearized to produce cross spectra. We also looked at other variations, other kinds of models that are included in DCM and how they differ and what different kinds of parameterizations they might have. Now, these models are operate at the mesoscopic level in the sense that they are an abstraction at the mesoscopic level which describe the brain's dynamics. And they link the macroscopic level, which is the global sort of brain dynamics, with the microscopic level, which features synaptic properties and receptors, kinetics, and so on. So the very intermediate link between these two very different scales and serve as a very powerful bridge. Next, we'll see model inversion. How do we fit these models to data? So we've covered the neuronal model. I'll briefly touch upon the observation model. Now, DCM can carry out simultaneous optimization of the spatial component, which is the forward model, the observation model, and the temporal component, which is the neuronal model. The observation model consists of this forward model, which can either be an equivalent current dipole, so the neural mass at a specific source is essentially a dipole, a current dipole. And this current dipole has 3 location parameters and 3 orientation parameters. If we are very confident about the location of our ECD, we can lock it and we need not optimize it from data. The other way is the imaging approach, which now has multiple dipoles all on the cortical surface. We have to choose for each source the radius of the sphere and the number of eigenmodes to consider across these dipoles within that sphere. Now, these both are for EEG and MEG. If we have already localized our data in source space, we can proceed with a virtual electrode or an LFP approach. Alternatively, we could directly also have LFP measurements, in which case the observation model is sort of a scalar gain function. Right. Now, as to the actual modeling steps, we start by first defining our experimental conditions and an input. In DCM for CSD, the input is colored noise, which follows a 1/f spectrum. Next, we specify a neuronal model, and this amounts to choosing a specific architecture of interest with a certain parameterization. We also select an observation model, which could be ECD, the imaging solution, or a virtual electrode or LFP. Next, we define our likelihood model and specify priors. Now, all the parameters in our model, they are log scaling parameters. So their prior expectations are 0. And we define variances, prior variances on each parameter, which control how much they can vary by being informed by data, right? And usually these prior variances come from physiologically meaningful ranges, so that the parameter estimates are constrained to something that is biologically plausible. Right. So now, if we have a lot of confidence in a certain parameter, we can reduce its prior variance, which means we are increasing its precision. So what we are saying is that now the data has to compete a lot more with our prior in order to shift the parameter from its 0 value, essentially shrinkage. Alternatively, we could be absolutely certain that this parameter must take this value and must not be informed by data, in which case we could lock its variance to 0. And in this case, the parameter will not be informed by data at all. Right, so once we have done that, we can invert this model, which amounts to estimating model evidence, also called the marginal likelihood. Once we do that, we also get posterior distributions of the parameter. Right, so now how does this actually work? We essentially start with our data, which is CSDs, and we take our generative model with a certain prior distribution. Now, these two are optimized by a Bayesian version of the expectation maximization algorithm called variational Laplace. uh, con- unlike sampling-based methods like Markov chain Monte Carlo, variational inference seeks to optimize an approximate posterior, and it does so by iterating over these two phases of expectation and maximization. The first is computing the expected log likelihood of data under an approximate posterior. The second is maximizing expected log likelihood with respect to precision parameters. So it alternates between these two steps and does a coordinate ascent until it reaches some convergence criteria. And once it reaches there, we obtain the posterior distributions of parameters, which we can use to carry out inference over parameters, such as asking whether a certain range of parameters is very likely or not. We also obtain model evidence, which is the variational free energy. And using this, we can perform inference over the model structure, such as should a parameter be included in our model or not. There are many kinds of such investigations that can be performed with DCM, such as we could compare whether network A here is better than network B with one less node. We can ask whether X and Y have a forward-only connection, so a bottom-up connection, or they have a top-down connection as well that is meaningful with regards to the data. We can ask which connections are modulated by experimental effects. We can ask whether an experimental effect is driven by extrinsic, that is between region, or intrinsic within region connections. We can also ask which neural population or which parameters within a neural population are affected by certain experimental effects. And a very nice example along these lines was sort of published recently by Amir Hussain Jafarian. Among other things, it also features an excellent reproducibility check where they consider odd and even trials separately and show that they yield similar estimates. Now, what they did was they took resting state MEG data, they fit DCM for CST using the same CMM NMDA model that I showed earlier. And they also separately obtained MRS GABA measurements for the same participants. And they asked which parameters in their model were best informed by MRS GABA. Right. So here is the model. More concretely, they created different models and they compared these models to identify which one had the greatest evidence and which one essentially had explained the data the best. So in the first variation, first model, they asked whether MRS GABA informs the recurrent or self-inhibitory connection of the superficial pyramidal and the deep pyramidal population. In the second model, they asked if MRS GABA informs the intralaminar inhibitory connections. There are three of them. Right. And for both these models, they also considered two different ways of mapping them. One was scaling it using a sigmoid. The other was scaling it with a polynomial expansion, I think second order. Now, so they have four models, recurrent versus intralaminar, and two variations of each of them. And they fit these four models and compared the model evidence for these four. And this is what they find. The winning model over here in the top plot is model number 2. The next one is model number 4, which is quite close. But the difference between them is about 20. And the difference of free energy about the magnitude of 20 corresponds to a log base factor of 20. So the model evidence is overwhelmingly in favor of model number 2. And that can be seen in this posterior probability of models. where 2 has almost 100% probability. So what this means is that a sigmoid transformation of MRS GABA informs the recurrent or self-inhibition of deep and superficial pyramidal populations. That is a very cool finding. And it was very nice the way they did it, because they split it into odd even and showed that, okay, this is a consistent reproducible finding. So we've reached the end of our discussion. To summarize, we've looked at the generative model in DCM. We built a neural mass model stepwise. We saw different variations of these models. We saw how they can produce power spectra after linearizing them and looking at their transfer functions. Then we saw cases where by varying certain parameters, we could produce specific changes to the power spectrum, basically forward modeling. We also saw a case where an example where these models were inverted from data, and the parameters of the model that best explained measurements were investigated. So all in all, this is a very powerful um, way of carrying out inference, specifically about mechanisms in the brain that cannot be directly observed or measured. Although these now inferences can be carried into further experiments, let's say in animals or in much more controlled studies, to specifically test if those predicted mechanisms behave the way they do. Well, I hope you try out DCM, and thank you very much for your attention.